So thank you everyone for joining us today. We're excited to have you for our 6th session in the CCMA webcast on image cytometry. So today we have an excellent group of panelists, as well as a presentation from Paul, who's at the Sunnybrook Research Institute out in Toronto. So we'll give it just a couple more minutes to let people pop in. And while people are joining, I will say thank you to our sponsors for today. We have BD as our series sponsor. And for this session, we have Luminix and TCAN as our session sponsors. So I think we've got the bulk of people in. So I will stop sharing and I will pass it over to Paul.
Okay, so I actually didn't create a presentation. I'm just going to kind of discuss what we do here, and thank you for giving me the opportunity to do so. So I am the manager of the Center for Cytometry and Scanning Microscopy, clumsily named the CCSM, here at the Sunnybrook Research Institute. I've only been here for about 2 years, but I have about a half of a career, about 14 years in cytometry, starting in 2007. Originally back in Ottawa, so I know Vera quite well. And anyways, and came to Toronto, did a little stint at Sick Kids, and then moved on to Sunnybrook Research Institute. So we have a really interesting facility here in that we have we offer a lot of services to what seems somewhat to be to a little amount of people. So that I think that's why we have everything kind of lumped into one facility. So what is in our facility? We have cell sorting, flow cytometry and analyzers, and we have some advanced microscopes. So we have a confocal microscope, a spinning disk microscope, and a traditional fluorescence microscope, as well as a microscope that has been augmented to look at calcium flux in neurons. So when I took this job, it was kind of the facility was introduced to me as sort of a mom and pop shop. We are, like I said, we offer a wide variety of instrumentation and training and education. So that instrumentation is 2 cell sorters. So we have a fusion and a backup ARIA 2. We currently have 2 LSR2s with different configurations, which many of you may know that 3 of those 4 instruments will be end of life in a year. So we are working quite hard. And, I was pleased to see the presentation that we had on funding sources, because we were working quite hard to replace the backup sorter and our 2 LSRs. And we are pretty close. I don't want to jinx it by saying too much more than that, but we do have a plan in place. Some of it is being enacted, but I am hoping that we have at least 2 new instruments within a calendar year, hopefully 3 or 4 as well. But I can almost certainly say we'll have 2. So that's very exciting times for that, to get in new technology, to expand the capabilities of our analysis. We do so, there's a lot of high-level immunology research happening here at the Sunnybrook Research Institute. So we're kind of limited right now with our technology. So right now, the maximum parameters we can do on an analyzer is 15 and 18 on the cell sorter. So, that's barely enough for the demand at this point. Anyways, and then there's also the imaging component, which was frankly relatively new to me. I've done a lot of fluorescence imaging and turf imaging in a much, much former life. So I kind of learned the confocal. We offer the confocal and the spinning disk, and I learned these on the job, if you will. Had a lot of really good support from Nikon along the way. And, now I would say I'm kind of middle of the road, let's say, for imaging. So lots of room to learn. But I do, appreciate, in the way I present this facility, is what we do offer at the end of the day is, fluorescence-based techniques. There is not that big of a gap between, fluorescence imaging, confocal imaging, and flow cytometry at the root of it. And that's how I tend to present it. And because of that, we have forged a really tight relationship with a neighboring facility, HICA, which is the high content cellular analysis facility. Again, high level microscopes, fluorescence based assays, and also have forged a really good relationship with a neighboring GMP facility. So we have a nice little pipeline that we're starting to form. So we have our cell sorting, our analyzers, our microscopes, which we offer access to, training, assisted acquisition. Our model is that all the sorting is done for me. Oh, I should mention, all of this is just me. There's no other employee. I am a manager, technician, gopher, at all levels for the CCSM at the moment. Although I am trying to slyly recruit some, backup sort operators and analyzer, anything, training assisted. Anyways, and again, because of the pandemic, we got to branch out a little bit. So we're making this nice little pipeline with GMP, with Flow, because they're using our facility, and I do also have a GMP background, so I can help them with some of their regulatory work. The hike is, again, just one step further down the fluorescence road. And we've finally saw some benefits, some rewards to this, and that we're starting to get a little bit into some contract research organization work coming out of the pandemic because of this pipeline that we can provide. So I don't want to say we're a jack of all trades, master of none. Maybe we are a jack of all trades, master of some in this facility, but it is the benefit of this mom and pop shop, a very intimate culture that we have here at the Sunnybrook Research Institute. So anyways, yeah, that's the kind of the story of the CCSM.
Awesome, thank you. That was a great summary, and hopefully that will spark some people to get in touch with you.
For sure.
So I'm going to pass it over to my co-host, Steve, now.
Thank you.
Get rocking and rolling on this discussion.
Okay, so before we get into the discussion proper, I'd like to take a couple of minutes just to let the panelists introduce themselves. So today we have Peter O'Toole from the University of York, Hartland Jackson from the Lunenfeld Tanenbaum Research Institute, and Elke Kuster-Schack from the Research Center of the St. John University Hospital, no, Saint-Justine University Hospital. And so what I'd like to do is just take it in turn for each of you to have a couple of minutes to introduce yourselves, tell us a little bit about what your current role is, and also a little bit about your background. So we'll start with Peter. So I'll turn it over to Peter. So Peter.
Steve, thank you very much. Well, my background is my wife's picture wall, and it's anyway. So I head the imaging and cytometry lab, a bit like yourself, Paul, up at the University of York in the UK. It is very broad. So we have the cell sorter, so that's an Astrios. We've got a couple of Cytoflex LX, Cytoflex S, Protester X20, a couple of Cyans. Then we've got the confocals. So we've got 780, 880, 980, two Aries scans, multiphoton, slide scanner. But actually for today, what's probably more important, when we think about cytometry, we're thinking about measurement of cells, getting quantitative information from cells. So we have something called a live site, which is a label-free quantitative phase image system, which is really cool, but also, and maybe of even greater interest, we have the NanoString GeoMx digital spatial profiling system that enables your 80 different antibodies simultaneously, or your 18,000, I think, is now RNA transcripts. So we're working on both of those systems. I say it's quite a broad lab. I've got electron microscopes as well, but they're not cytometry. They're a dark art. There's not even any color in them half the time. So that's my day job, but I also head all the core facilities at York for the life sciences. I wear a hat for the Royal Microscopical Society, and we do loads of courses and lots of other bits and pieces. But anyway, sorry, I will stop there and I shall pass over. Thanks, Steve.
Okay, let's hear from Hartland next.
Thanks, Steve. So I'm a pretty new investigator at Lunenfeld Tanenbaum and part of the Department of Molecular Genetics at U of T. So I'm in Toronto. My past history, I was lucky enough to be at the University of Zurich and work alongside some of the creators of imaging mass cytometry, and then be along for the ride to guide it through to kind of a commercial product and see what we could do with kind of high dimension imaging using imaging cytof. So the difference in this technology is that the antibodies we're using have metal isotope tags instead of fluorophores, and we're reading them out with mass spec. So now in my new research group that just started kind of mid 2020, we're now focused on this technology with my own team. So we have our own imaging CyTOF unit, but then we work closely with the other services within the Institute. We're combining multiplexed imaging with a lot of other things. I think the base thing is always be slide scanning up front, combining with fluorescence. We're looking at other multiplexed fluorescent-based imaging methods, their combination with this, and also some of the technologies that Peter mentioned in terms of some of the spatial transcriptomics and how to combine different image cytometry tools, whether that's on the same sample or in serial sections to provide different benefits of different technologies to elucidate the cellular content of specific features or pathology features.
Okay, thank you. And then finally, Elke, can you tell us a little bit about yourself?
Hi, yeah, thank you for inviting me here. I am running a small facility, well, small in terms of the staff, that's just me, similar to what Paul was talking about. My facility is a platform for light microscopy, and I run 14 high-end light microscopes of different flavors. I was surprised and honored to be part of this panel, and I had a hard time kind of picturing me in this cytometry field, but then I realized that what we do when we do quantitative imaging, as we call it, is actually cytometry. We measure things about cells. And one thing that I always see myself working with the users of the facility with is going from using microscopy as an illustration tool to actually capturing data. And then when we use methods that are more high throughput, like my slide scanner, actually going from looking at a couple of what is previously or going in considered typical areas of the tissue or typical cells, going towards imaging everything and measuring everything and putting it in context, which actually ties in with my background. I actually, all my research training was in protein structure, biochemistry, protein interaction, and I still cringe when people ask me about co-localization, because two molecules cannot be in the same place at the same time. They just can't. So that's where I fit in, and I hope to kind of look at things from this side. And I'm relatively new to microscopy, actually. I worked with other technology in a core context before, but I saw the light about 12 years ago, and there was no going back.
Okay, thanks very much. So you've kind of echoed my mantra, which is that images are not really pictures, they're more or less numbers. And if you can start thinking about them as numbers, then that really sets you up quite nicely to think about what you can measure about them downstream after you have all these numbers. So with that, I think we'll kind of kick off the discussion with a little bit about, I mean, most of you, or all three of you have answered this to some extent already, but I guess the first thing that we'll kick off with is what image cytometry means to you in particular. So since you're already on the screen, Elke, why don't you start? Why don't we start with you?
Well, I already started down that road earlier, where I said, okay, so image cytometry initially just means I'm trying to measure something about cells from an image. And as you said correctly, an image is just, I mean, I always say it's a spreadsheet of numbers. We have intensities over space. And I think one of the things that I'm only starting to really appreciate is that there's so much more in our image information than we usually set out to measure. So yes, we might be using this one antibody to see where this thing is, but along the way, we might actually also be able to look at the shape of the cells, look at the volume of the cells, look at other factors. And that is where newer tools that allow us to look at lots of data at the same time, thinking about deep learning, artificial intelligence, really come in to allow us to extract all that information. So that's where I see microscopy with cytometry, like the overlap.
And panelists, feel free to just jump in. This doesn't need to be like an organized discussion. So
I can reverse the order and keep it going. I think okay, nailed it pretty succinctly for me by saying it's kind of measurements in space, and specifically around cells and looking at the spatial organization of different things, whether that's subcellular features or whether that's different cells and how they're organized and interacting with each other and making up a tissue. And I think that's kind of different aspects of this is what we're all trying to get at with different imaging technologies, and kind of hopefully all trying to push to what information is extracted, as Elke mentioned, not just the one marker, but all the other features that are in there, and what can we do with that is kind of the fun part.
oh yeah.
If I can add to that, I think imaging cytometry, you've heard a lot of different applications. If you've listened to what you all have here, will all be different.
Mm-hmm.
And actually imaging cytometry will be a different tool for different users. And it's very much looking at when a user comes in, what they need out of their data. And that, I guess, that's where our specialism is. You know, us at Core Facilities, we're the ones who are to work out the best cytometric approach to take those measurements for the user. And quite often there still isn't one tool that addresses all their needs. And it's combining those combinations. And it's great to hear so many have got microscopes and cytometers, because they are so entwined. And I think actually when it's in one lab, it encourages users to take their cell sorted samples over to the microscope, or when they're trying to do quantitative cytometry using a confocal, going, no, just shove it on a flow cytometer. And you get different answers. And we've got the niche, we've got the really high end stuff, but the basics, the users still maybe need encouraging sometimes to realize they're not just pretty pictures, but as Elke said, you know, it's getting that extra information out of it.
For sure. And that really leads into one of the big things we wanted to discuss in this panel is how do you overcome some of these barriers or limitations for users? How do you convince them to pursue image cytometry when, say, they've been a flow cytometrist predominantly, or a microscopist looking at other image cytometry techniques? So what are some of the approaches you take to address these limitations or barriers for the people you work with?
If I can just chime in here, it helps for me when I start talking to somebody in a consultation prior to deciding which technology they want to go into, to also make them aware that their approach is guided by prior limitations. So they might come and say, oh, I want to replicate this that I saw in this publication with my cells. So we have to use a 488 nanometer laser to do this. And then I step back and say, okay, let's just look at the methods and materials here. Maybe these authors only used the 488 nanometer or this filter or something because that was the only thing they had. So I'm trying to really make people step back from all these preconceptions and really get to the pure question that they're asking, so that we can then, you know, look at the platform's offerings or beyond. I mean, here in Montreal, we're really lucky to have a good communication among the core facilities, so that we're kind of aware what is available in other facilities. I'm located in a hospital research center, but that doesn't mean that people can't also access things in other places. So like I say, extracting the actual question and then trying to just instrument and the technology agnostically, trying to find out what is it really that you want to know about your thing, and then you can actually think about the technology that might get you there.
I think that's a really important approach to take. Is it's not just I want to use this shiniest, fanciest tool. It's I need to figure out what I want to know from my cells, and then from there direct to which instrument is going to best allow you to capture that information. And I find that's something that I have a challenge with in my facilities. People tend to want to jump on the new buzzy technology, even though it's maybe not well suited for what they want to look at.
I think I also live this problem, and maybe I'm even talking some collaborators out of working with me as they show up, and I kind of dive into, well, what do you really want to know? Or do you just want to do this fancy method? And then, the questions come around, and then I might push them to someone else to be doing different approach or different types of quantitative things. I think you both kind of nailed it. It really depends on the question.
I think one of the other parts of that question that you asked to start with, how do you get users to engage with different techniques as well? And one of the best ways we've found to do that is, besides one tactic is to hijack someone's samples and try something different with them. But we've also used studentships to great effect in internships. So in the UK, some of the PhDs have to do lab placements outside of their main lab. And so we kind of use those to try some pilot studies. So it's lower risk for the academic to try something that isn't their main field of getting results, so we can exemplify it. The other bit that's really good is user group meetings. So we get users to talk at user group meetings. If you have a PhD student talk, they will bring their mates with them. So it kind of diversifies who is attending those meetings. And if they're talking about something new and exciting, more people get to hear about it. And actually hearing a user doing it makes it far more approachable than us telling them that they could do it. If we can do it, it must be really hard. If a user can do it, well, that means they can do it as well. So we find that as a really good way of choosing different technologies and getting a new user, a user that's done something new and novel, to talk to the wider audience to show what they can do.
I think we all kind of sit also in the imaging field where seeing is believing sometimes. And I think we all mentioned there's this kind of easy first step of creating beautiful images, and that sometimes gets people hooked. And then they realize that the bottleneck may be more at the data analysis stage and actually extracting, turning this from qualitative, beautiful images to actually quantitative data can be a big step, but they're already hooked because look how beautiful it is, and look how nice. And I want to sit at this scope, and I want to do this method.
So you mentioned data analysis, but I think a kind of more, not important, but more fundamental part of this problem is data management. Now we're all dealing with 800 protein localizations or 80,000 messenger localizations of all these cells. This is now a different kind of scale of data that we're all dealing with. It's not just a single 512 by 512 image, but now thousands of images or tens of thousands of images. And so I'd like to hear from you how you deal with the data.
I'll have the worst answer to that. Our central IT has all the storage and the IT compute power. So we default to the central IT because we're not unique. As I say, if you do a light sheet microscopy experiment or a super res, or even the label 3 in the early days, you could be turning out a terabyte overnight. So actually the genomics lab for their single cell, huge amount of data, the mass spec imaging, huge amount of data, the cryo-EM, huge amount. So actually, instead of solving it ourselves, we put it out. But then it's the users who have to look after that data. And that's maybe the biggest risk, is the users themselves. We delegate to them to look after it on this resource. And hopefully we've got enough compute power for them to access the server licenses to actually do the analysis, which is probably the harder bit. It's less the storage, it's how they access the analysis capabilities.
I think maybe you have a unique situation where your IT team is maybe integral to the research, basically, but I think that will vary at many institutions. A lot of IT support is basically, here's how to do Microsoft Excel and Word. So I think that can vary across institutions. Yeah.
So we were like that, and we did have our own. So in the technology facility, we had our own IT team because that's how we started off. But university got quite wise to this and realized that, biology, chemistry, physics, psychology, they all have the same needs. And we were spending bucket loads of money inefficiently. And so actually we championed back to faculty and the university and said, well, this is just a nonsense. This isn't our expertise. We're replicating staff. And so, yeah, so it is IT services who I'd have asked how to install the latest version of Windows are now the ones who've got the expertise to manage our data.
Mm-hmm.
Fairly well.
Well,
Better
you're
than I
lucky.
could do,
I mean, in our institution, I also, I push the responsibility for the data to the user because I'm just myself with these 14 microscopes and 17 computers. I do have IT support here in this hospital research center, but most of their answers are around no. We cannot do that because we are part of the hospital network. So we cannot, for example, upload to just any cloud server. We can only do that to OneDrive because all the other ones are insecure. I pester my users with the fact that I don't allow USB drives on the machines. I mean, the data output on our machines is still small enough that it could be done to carry it away on a hard drive. But I also got viruses that you can't get rid of with some hand sanitizers. But it is a real problem because, as we said, the data amounts get big really, really fast. It's not just a question of the size. Just this morning, I tried to help somebody where apparently the data integrity was also compromised in the process of them trying to store it and then move it into a place where the analysis takes place. So it's a real catch-22 where we're trying to, at the same time, keep our computers functional and keep the data off of them, but also help the users to make use of that data in some useful way. It's not, I haven't found a good answer for it.
I think I'm kind of in the privileged position where we get to be pretty focused on one technology and kind of an individual data type and analysis pipeline. So I don't have the same issues of dealing with 14 different types of file structures and things like that, which is nice. But on the other side, we really see kind of the data processing and the data analysis pipeline is kind of part of the experiment, and it kind of all needs to be one piece. So for this purpose, we do have really locked how you'd have a wet lab protocol. There's a pretty standard data processing protocol that has specific scripts. Those are all backed up and saved. And then you're also saving the scripts for processing and early analysis steps with your actual hard data, and you're backing that all up together. So that then in your backup years from now, you have the raw data, but you also have what's going to produce the data types that you need to complete the analysis, and it's there. And you save space because you don't save all these intermediate steps. You just save the code that's going to reproduce them for you if you need. That being said, the pipelines are also changing all the time. You're trying to keep up with everything. And so you do kind of have to make it modular and pop pieces in and out and test them and learn where the flaws are and all these things. But I think having the IT support and data structure to actually extract information from these technologies is absolutely crucial. Until you can take it to somewhere that you can run statistics and get specific answers, it's nice, but you have to get to an answer. Otherwise, this is something to teach all your students too, right? You can go produce all these experiments, but unless you're getting something out of them, then you're not done. And it's not there yet. So it's kind of tie it all together into one.
I think Hartland is putting this in an interesting perspective because he brings up the analysis part. So IT to me has definitely at least two aspects. There's the pure infrastructure of having enough space, having the lines to connect one place to another. But then there's the other aspect of what to do with all this data and how to analyze that. And as a core facility that has all kinds of users, I'm kind of in a hard place because there are no cookie cutter answers to any of the questions that people come to me. I mean, even if somebody comes with an easy question like count these cells that were all labeled with DAPI for the nucleus, I don't have just a program where they can run their stuff through, because it might come from all kinds of different instruments. I have very open analysis software, but that means the person has to then use that software to actually build the analysis themselves. They can't just push the button that says count all the cells. And on the one hand, I love to be in that position because we're very flexible and we might use these tools to do all kinds of things, but that means the users actually have to come up with all these pipelines themselves. And that is not always, well, that's not always possible for everyone, because not everyone can go that route. So I'm not really sure what is the answer to that problem. So how do you actually help people go that way without actually having an analysis expert at hand?
I think the analysis is the toughest part. There are labs now who are having analysts in their lab, which can help with that. So we've got Karen, who helps with the more technical, TSNE type flow analysis. I've got Grant, who can help with the digital spatial profiling, so the high-plex. And we've got a couple of the team that can do the label-free, but we don't have time to do their analysis, and the analysis is not straightforward. So there's a lot of investment in training the users how to use it. And we do get good commercial support. So the companies who are providing it do help with training as well. And I think we have to lean on them for that. It's a really hard to cost recover analysts in the lab. That it's not something people instinctively put on their grant. I'd just like to do one caveat to what I said earlier. When our PCs are about to break, we go and buy a remote hard disk and back everything up just in case users haven't done it right. So users don't know that. But so we actually go back to the Luddite approach, just belts and braces. We're not really that confident in everything.
Cat's out of the bag now, I guess. Secret's out.
I just checked with one of our users here. It's fine.
Excellent. Good. We'll try to keep it that way. So we just had a question come in that I think fits into this quite nicely, and talking a little bit about budgeting for new equipment. And when you do this budgeting or purchasing or, grant applications, do you budget in data storage? Because having these appropriately sized servers can get quite expensive. So I guess maybe not so much for you, Peter, because you have an IT department that seems to be.
But they have to add their costs. Have to add their costs onto the grant for that storage space, and I don't think I always remember to remind them to do that. So actually, that's a bad on my behalf. I don't always remember to tell them to do that, and I should.
Well, and it seems like something that needs to be at the forefront of more of our minds, because I know I don't think about it all that often either, but like even flow cytometry data now is getting larger and more complex. It seems anything in this cytometry world is really requiring this extra data storage.
So from a Canadian perspective, every year I'm writing to Compute Canada to get access to resources for both analysis on their servers, but they also can provide backup services and backup to tape. So longer term backup as well. And so that's kind of a just annual maintenance and upkeep is writing applications to them. And they provide a very crucial resource, I think, for scientists everywhere, whether that's access to computational resources or support to get access to the cluster and servers and things like that, down to data backup. And so we end up doing some ourselves, and also then having the longer term backup go to them. And we've set up automated systems to make sure that it's running every week off of our acquisition machines. So if you do have IT that can help you set up things that have, and we just have a little thing that checks, is the acquisition software open? If it's left open, you don't get a backup, but if it's closed, then it will automatically back up every Sunday night, kind of thing. And then you make sure people don't produce more data than they, unnecessary images and things that they don't need. And then otherwise that backup keeps growing exponentially.
Yeah, I think you're a great example of somebody who is definitely very keenly aware of the problem. in my role as a facility manager, I have to kind of bring that message to a lot of PIs who are not aware, and send their students with a little data stick saying, okay, this is our approach to keeping our data safe. So just like with the approach to imaging and asking questions, we actually have to go back a couple steps from the point where somebody enters my office and says, I want to use your microscopes, or I want to count cells, or I want to do some other measurement, and make them and their PIs aware that there's more to that. And sometimes it's hard because I'm not in the communication loop as a non-faculty person to bring that message out. So I have to rely on my scientific advisors who are PIs to actually get that message out, so that people do ask for these for funds for these resources, for equipment. And I mean, just like some PIs still purchase instruments without service contracts, others purchase instruments that generate tons of data that and don't have an idea where to put that data.
Can I just add, I'm just reading the chat of what people are putting in there as well. So I think even Google, you can store huge amounts of terabytes of data for free. But it costs the user then to recall it and to access it back. So it's a really good way to sort of cost, we default, it doesn't cost us anything, but then the user, if they need to call it back, it will cost them to draw it back off at that point. It's not a huge cost. Something we haven't talked about is new analysis approaches. We're all talking mostly about off-the-shelf approaches, but when we're talking about imaging cytometry in particular, I don't think we're doing a lot with the data. It's so data rich, we're still asking fairly binary questions with it. And something we're trying to do at the moment is invest with PhDs and postdocs to actually now start developing new tools to analyze the data. And Elke, you mentioned deep learning, machine learning, artificial intelligence, all kind of the same thing in a way. It's really now how we embrace that, and how do we get computer scientists to be interested in our data sets to start moving that forward in a more meaningful manner.
Yeah, and that's a really great question. And it seems to be almost necessary for moving the field forward, because the data sets are so rich with information that, like you said, we're barely scratching the surface of them. But I mean, as a biologist, I do not possess the skills to go in there and do the computation necessary for those types of analysis. So have any of you been successful in bringing bioinformaticians or other machine learning experts into your area to work with you? And how have you taken this approach? Or is this something we need to figure out as a field?
Well, I'm only taking baby steps myself, kind of immersing myself into the ideas. And there are some amazing groups out there. There's the Henriquez lab he's now in Portugal, I think, doing some development towards making this available for no cost. So using some of these resources like the Google Cloud, I think, so that people can actually try it. And since I'm only learning myself, most of my users are less kind of imaging savvy when they come in. It would have to be me bringing that idea to people. So until I understand it, I don't think they're going to try it. But I think this is the way things go, that these things, I mean, while they're being developed in specialized labs, they're not accessible to people like us. But I think this is just on the verge to get accessible for people even without a specialist in place. And that's a really exciting thing. And I try to, then I try to identify people, users here who might actually be of the type who is interested in trying something new. I mean, to me, many of the users fall into broad categories, those who like to work with established protocols and those who would also like to push the envelope a little bit. And that, I mean, the AI approach is certainly something that I can't try alone because I don't generate data, but I will find, I hope, some people who want to try that with me.
And so, yeah, Ricardo, by the way, Ricardo is super cool. He is very entertaining. Ricardo Enriquez over in Portugal. But I think the biggest challenge, they're developing great tools, and actually Nature Methods, the last couple of versions had a lot on data analysis, but still so much of it is segmentation limited. There's no time resolution, and it doesn't do the big high-plex stuff still for the multiple colors. And my PhD, one of my PhD students is actually a mathematician, comp scientist, because I think it's easier for them to learn biology than it is for biologists to learn the comp science maths, and we need those skills. It's amazing what they don't learn in biology, but they can apply their skills. And actually a PhD is a great way to go, but you need to look for collaborators that are keen to solve these types of questions, because just because they're a computer scientist doesn't mean they're interested in our data sets. Just because they do image analysis, they're looking at people walking around streets or in and out of cars. Oh my God, cells are so much more different. When they actually look at the data, it's really complex. But maybe it is PhDs that are the way to go in collaboration with someone, and give them easy targets so they can do so, they can get a thesis out of it. But I think that's the way it will go over the next few years. But yeah, open source, I think it's the only way it's going to make it widely applicable in the early stages. And look at APEER, is it? I think APEER, which puts together cell profiler, Fiji, stroke image J, stroke C, stroke R code, and they can string those things together, because quite often there's not one solution for people doing that high-plex imaging cytometry. It's beyond the basics.
I definitely agree. I think this accessibility issue is a major thing. And I think that's, you kind of want some of these technologies to move through to be where the analysis is click button solutions. And I think the people willing to spend the time to create them is not quite there yet. Whereas I think as soon as, there's a way for any user to come in and immediately do flow cytometry style analysis off of images, that'll be a big breakthrough for all of those technologies. I have had lots of opportunities to work with fantastic computational sciences. I think benefit for me is I'm always trying to entice them in and bring in other PhDs that are the computational specialists by providing them something they can't get somewhere else, whether that's a type of data or larger data sets or clinical samples, and try to actually do projects together. I think a lot of the time people produce the data and then they go to the computational team and say, hey, look what I got, fix this, deal it. Somehow I've ended up that person occasionally now as well. And I'm not the person that you should be going to for that. But I see the struggles that the computational people have if they're not there from the forefront to understand how the data is produced and to understand the steps in processing that are necessary and actual, that it's produced properly and reproducibly so that they can make these tools. So we do now, we do see a lot of potential in image cytometry for new types of analysis and new methods. And I think, we're lucky that we're in this era of single cell obsession, and that, so once you can add in the buzzwords, people jump in. But we work very closely with Kieran Campbell's lab, who's another new investigator at Lunenfeld, an excellent PhD student, Shanza Ayeb's here on the call. She's doing amazing new things with imaging CyTOF data. So basically one of these people that Peter is describing is luckily here, seeing what I'm talking about, knowing that I'm going to show up on her committee in a couple of days and ask her all the sorts of questions about what I'm saying right now. That's not a threat, I promise. And but otherwise, I think it's, these are collaborations. There has to be something there for the computational people as well, whether that's they need to be enticed by biology or the problem. And sometimes their problems are different. They want a technical issue or something like that. Peter?
Hartland, just to back that up, I think one of our responsibilities is to shout at our funders and let them know that this is needed and that they should be investing in, postdoctoral positions and fellowships and potentially academic careers in this area. They're all telling us that AI is the future. And they've got to invest in people wanting to do the AI to address our needs, not just genomics. Okay,
that's true. So even for cell segmentation, there's been big breakthroughs in the last year of tools that are, aren't requiring weeks of my time to produce training data, and are BAM giving us cell segmentation and single cell data, and that's moves the field forward really a big giant step. And I think there's still a lot of places that this can be improved. I think in terms of funders, as you're mentioning, there's more coming out now, and I think like CZI or Chan Zuckerberg is really leading the way to actually provide grants that are just for software development, or only for, to provide these tools to the community-based tools that, they're only there. There's a whole grant just for image specialists who are going to be highly community involved and build new tools. And I think these things, they're not going to provide you the one flashy thing that most people need for the grant, but they're going to contribute to the community and have a ripple effect that's going to have a much bigger impact. So I think as one institute's doing this, I think more will see the benefit of it, that more will come, and we can keep talking about it as well, Peter, and pushing for what we need.
So how do you champion directly to your funders? How do the funders know what you want or what you need?
If you're directing that at me, that's tough.
No, definitely not. Across the board. The UK, we have the facilities meetings, a bit like this, every January. But the funders attend it. So we have both the charity funders, so Wellcome Trust, but we also have the government funders, so medical research, the biological funders, and they come to the meeting and hear what we need. And we sit on their strategy committees, telling them what we need. Keep chipping away at them.
Yeah.
And they start funding it.
So here in Canada, an offshoot from the CCMA developed, which was the CNSP, or the Canadian Network for Scientific Platforms. And they've kind of taken that role, but championing it from instrument procurement almost exclusively. So I think that would be interesting to try weave this into studentships, because right now they're just dealing with the CFI, the major grantor for infrastructure. But if, like you said, they started bringing in the other major funding agencies, there could be opportunities for studentships, because this is really a big need in the field.
I think there is also an ongoing culture shift in turn. There's been a bit of a pushback on this flash pizzazz type publications and the research grants requiring just extreme excellence and always wanting to do this super hot thing. And I hope that comes around, but from my experience in the first years, putting in grants that I think are just for required necessary tools, the response is, yeah, this is nice, but this isn't excellent. This isn't mind-blowing. Yeah. So you're going to get money. You're not going to get funded. And so, I'm constantly trying to tuck these things in as this is required in order to do this next step. And unfortunately, that's a strange place to be, rather than just being very direct and saying, everybody needs this. This is going to help everyone. Let's make this simple goal. I don't get the grant then. So I think as it comes around, and as the message goes out there, and as more people run into these technical challenges, I hope those people end up on the grant panels, and there's someone there to fight for the practical aspect of things as well. And I hope it happens.
Yeah, I think you're touching on a very important aspect here, that we're breaking down these barriers, things that used to be in these nice little silos. Okay, here are the biologists doing their thing, here are the IT people doing their thing, and they have nothing to do with each other, and they each write their own grants, and they have nothing to do with each other. Everything weaves into each other, into into everything else. And just like on this panel, we bring in all these different perspectives under an umbrella that we didn't even think was there. That has to happen on so many different levels. And as we said in the beginning, we have to work with the users of the technology, trying to open their mind to the opportunities in their samples. But we also have to work on the other end to actually bring the opportunity to use this technology into the labs.
Perfect. I think that is a great note to kind of open up now for any questions from the audience. So a few have come through the chat, and I think we've addressed most of those. But if anybody has questions for our amazing panelists, please feel free to unmute yourself and ask away. Otherwise, while we're waiting for questions to come through, how about we just go with like a general question is, where do you see the future of image cytometry, say in like the next 5 years? Where do you see our field sitting?
Didn't we just answer that in the image analysis?
I think maybe.
I think it's really scary just how fast the field is moving. The nanostring was the first big genomics multiplex side, but you've got Codex. None of us talked about Codex or Akoya or, oh gosh, what are they all called?
I know there's so many now. There's so many.
This is kind of where I sit. And so, yeah, so I'm even like, I've come up with one technology, but we are trying them all and comparing them all and seeing which one is going to have life. And they all have their own benefits and trade-offs as well. I can plug, I have an older webinar for Abcam that's up on their website where I just talk about all the different multiplexed antibody imaging technologies and how they compare and how you're going to get bigger area with this one, but you're going to get more markers with this one. And I think it's really what it ends up coming down to is who's going to make it good, easy for the end user. That's the one that's going to, I can pull one. Yeah.
Yeah, put a link for that.
So who's going to make it easy? Who's going to actually provide, here we go. Who's going to provide the whole user interface, right? Like, I think there's a whole, we didn't even touch on antibody field. That's a huge conundrum of which ones are actually specific and which are working. And if you want to go, now you're doing 40 marker stains, you have to validate each of these independently and the work behind that. And I think, now each of them is also building in transcriptomics. There's going to be spatial transcriptomics that's going to come. I think you're going to be wanting to do multimodal. It's going to go where single cell has been. So there's going to be proteins and transcripts at the single cell level. What's that going to cost? How much area? How many can you do? What are the issues is going to come along? But these things will happen. I think there will be spatial sequencing as well at some point. In situ sequencing will come, and we're going to be doing image cytometry for actual individual oligos and RNA transcripts. And Peter has even more things to list, I'm sure.
No, I was going to add that we're already correlating, so taking parallel sections for spatial transcriptomics, spatial phenotyping with your 80 antibodies, and then mass spec imaging of those to actually get the metabolomic data sets. I think the big thing for me is I want one instrument that does it all. At the moment, you can do something with subcellular, something that does big regions, and they all have their pros and cons. So actually on Hartland, I have already clicked on your link and I will look at it and watch it. They, as you'll hear from Hartland, I'm sure they all have their pros and cons, and there's no one solution for everything yet. And we want to do proteins, they'll want to do RNA, they'll want to do sequencing. The biggest problem at the moment is the cost of the reagents, because actually when you think about 40 antibodies in one experiment, it's no wonder it's so expensive. But that has to come down, because that is a hugely prohibitive step for many researchers, because you're talking maybe UK £10,000 to do an experiment just to see if it works. It's a lot of fun. For some people, that's nearly their annual consumable budget on one experiment. We need a technology that makes it cheaper to use.
Peter, I'll interject there for the cost of a 40 panel antibody. You already mentioned, or Hartland may have mentioned it. It's the validation of those 40 antibodies, each and every vial that gets sent out. That's why it costs so much. I mean, we're building a small 3 to 4 panel antibody selection for small particles, and trust me, it's expensive.
Yeah, no, I know exactly why it costs so much.
Yeah.
I'm not questioning why it costs so much. And just think about it. If we were to buy 40 independent antibodies, it would still, it's actually, put it that way, it's cheap. It's not that expensive. We just need to find a way to make it cheaper, because when it's cheaper, the market will open up to so many more users. And then all these analysis solutions will be found because there'll be so many more people wanting to get access to it, asking more difficult questions. It all comes down to the breadth of user base, which isn't that big at the moment because there's only so many that can afford it.
I agree. No, I agree.
But if we could get that cheaper, so what is the probe type? Is it going to be an aptamer type solution that doesn't need validating because of cross reactivity? Maybe that would do it. Maybe that would just unlock it all. I don't know. There must be some way of doing it. Must be.
I think if you figure that out, you're set.
I think so. Any other questions or comments from any of our audience members? I don't see anything really coming up in here. Any last thoughts from any of our panelists? Otherwise, we're pretty much done for time anyways.
Yeah. I will say one of the things that you guys brought up, we've been very lucky at actually bringing in co-op students from the different computational areas, whether it's machine learning or whatever. And that's actually worked out really, really well. Very specific questions that we give them to do. But typically they come in, they see the data set that we have, and they want to try so many other things. So we usually get not a 2-4, but we get multiple channels going, and they sometimes come back, and we've had computational scientists that have stayed with us for 6-7 years now. And
we need to start submitting these data to those hackathons that they have.
And we do. We actively play in those.
Perfect. Well, on that note, thank you again for our excellent panelists for joining us. Look forward to working with you more in the future as we figure out all of our image cytometry issues and data pipelines. And thank you again to our sponsors. So BD as session sponsor and Luminix and TCAN as our series sponsors. And we hope you will all join us for our next session sometime at the end of November, where we'll be talking about facility management software and looking at choices and how to implement them into your facilities. And on that note, thank you so much for joining. This will be live on our YouTube channel probably later today. So if you have any questions or comments that come up later, feel free to post them in the chat on the video, and we'll get them addressed for you. All right.
TCAN introduces Spark Cyto, the first multi-mode plate reader with real-time cytometry and imaging, the perfect research tool for multiplexing and live cell analyses. Spark Cyto offers full cell incubation capabilities, controlled for temperature, gas, and humidity, monitors cell growth, viability, cell death, and other relevant parameters across the entire well. Never miss a critical biological event again. Learn more at tcan.com/sparkcyto.
The image stream is a high-throughput, high-data content imaging flow cytometer. We have recently released high-gain mode. Its superior photonic sensitivity makes it ideally suited for imaging small particles and extracellular vesicles.